everybody. Now, we are seeing our research. Neodymium oxide nanoparticle produce toxicity and cation cerebrovascular Zebrafish Embryos, while Apoptosis Pathway.
30 well-developed zebrafish embryos were placed into each well and incubated with neodymium oxide nanoparticles at different concentrations after laying eggs for 10 hours post-fertilization. Then we observed the morphological change by microscope per 24 hours until 120 hours post-fertilization. We found the mortality of zebrafish embryos gradually rise in a dose-dependent way. After 5 days of treated, we assessed cardiac performance using confocal real-time recording. The heart rate slowed down and was irregular in treated embryos. The live embryos were anesthetized with tricaine and mounted in 1% low melting point agarose for imaging with Nikon A1 Plus confocal microscope. In our research, the most interesting thing was the cerebrovascular vanished in embryos exposed to neodymium oxide. We used the FLIC1 EGFP transgenic embryos, in which the EGFP labeled all vascular endothelial cells. The cerebrovascular was intact on 3-day post-fertilization, but the disappearance of cerebrovascular happened in 4-day and 5-day post-fertilization. To our surprise, the cerebrovascular arrangement of treated embryos was disordered on the 3rd day. In Y-type embryos, the vessels showed like pipes or lines, while the vessels showed more discontinuous dots in neodymium oxide exposure embryos. Then we want to explore further the mechanism of toxicity. Why caused the blood vessels disappearance? The first thing that came to mind was to detect the apoptosis and autophagy pathway. The results of immunoblotting showed the autophagy-related protein LC3 was not changed, but the apoptosis markedly increased. Although we observed the change of protein levels, it cannot explain the relationship between the two pathways and the cerebrovascular disappearance. So we injected the mCherry LC3 mRNA into FLIC1 EGFP transgenic zebrafish embryos. We found the abundant LC3-2 puncta in EGFP positive cells. The cerebrovascular was decreased compared with the control group, indicating the autophagy downregulated in the cerebrovascular when neodymium oxide treated. The tunnel staining indicated that embryos exposed with neodymium oxide nanoparticles have marked increased red spots on the head. To sum up, neodymium oxide can induce toxic effects in a dose-dependent way by activating apoptosis pathway. The downregulated autophagy flux in cerebrovascular vessels might affect vessel sprouting and contribute to the vanished cerebrovascular. This study hoped to provide practice guidance for minors, workers, especially children and pregnant women, related to nanomaterial. And this study might provide instructions for comprehensive evaluation on occupational health. In the end, thank you for listening, and also thanks for the help of our research group.